Well, I will get started. Thank you, everybody, for joining us. This call will be recorded and available shortly after the meeting. We are going to have a full hour with lots of great information covered, and we will facilitate a question and answer at the end. So any questions that you have, please add those to the chat, and Dr. Dylan Verden will collect those, and we'll do that at the end. So today I would just like to review our agenda, and then I'm going to hand it over to, we have two presenters today that I'm really excited to introduce. So our agenda, we will review the ASCEND study, the KOALA study, blood collection, current ASOs being used, and information from n-Lorem. So, of course, our first presenter today will be Dr. Wendy Chung from Boston Children's Hospital, and she'll be covering all of our natural history studies that they help us facilitate. And then we'll hear from Dr. Sarah Glass from n-Lorem, and we're really excited to have both of them with us. I know they also have some team members, so as you guys start talking, maybe you could introduce your team members if you know who's on the call today. There's so many people behind the scenes that go into all of this, and we appreciate you all. So I will facilitate the PowerPoint. So Dr. Chung and Dr. Glass, if you'll just let me know when you want the next slide, I'll hit forward.
So I guess I'll go first, Angie. So thanks for that. Just to let you know, we've got, as Angie said, lots of members of our team behind the scene: Dr. Sunawa, Dr. Rincon, Dr. Zakara, Joanne Carroll, Jessica Waxler. Many of these are people that you've met before, some also new to the scene, but all of these working both at the bench as well as working on the clinical research that I'll be talking about today. So in terms of this, and let me try and be clear, as Angie was saying, there are two different studies and they're all related to each other. I'll try and explain the method in terms of the reasoning behind this. The first is that we wanted to be as inclusive as possible and to leave no one out of this. And in doing this, based on our geography around the world, we know it's hard to travel, especially for individuals who may have medical issues. And so ASCEND is an online study. Within this, ASCEND is, again, we tried to make this open and available for individuals so that we have interpreters that can handle multiple languages. We realize that may take a little bit more time, and we appreciate your patience, literally being able to do the translation with this. But as we're doing this, the reasoning is to be able to get data for individuals over time. And you might ask, well, why do you need the information? And I would say there are multiple reasons. Number one is we want to stand up and be counted. And so when it comes to individuals who try and say, how common is CAND, or KIF1A associated neurological disorder? And I will be honest, people, are trying to decide how much money to invest in developing treatments. We want to be able to say that this is more common than you think it is. And so we want to be able to stand up and be counted in terms of that. We also want to be able to, I think of it as the care until the cure. And so as we think about this and think about our care and standards of care and being able to have evidence to say that there are certain things that your doctor should be doing to take care of you or the person in your family, we need to have the evidence behind it. And as things are published in the medical literature, it develops the evidence to say to doctors around the world that this is what should be done. It also gets insurance companies or payers to be able to pay for therapies, for medications, for surgeries, for whatever it might be, but it produces the evidence to be able to get to that. And so with this, we're trying to be rigorous. We're trying to do this in a very methodical way in terms of understanding this. And it's complicated because, as most of you know, this condition changes over time. And so it's really important to be able to get longitudinal data over time. And I just realized I'm probably speaking quickly, so I'm going to slow down. As we try and get information over time, We sometimes see a phase where children develop, and so they gain skills and they gain abilities, but then for many of them, they turn a corner and they may actually decline. And it's important to be able to understand that and understand what might be changing, literally from the head to the toe, and to be able to understand that comprehensively. And importantly, understand if there are strategies, therapies, educational tools, assisted devices, surgeries, whatever they might be, but things that will help. And so as we're doing this, we're trying to make the most of every single person by squeezing as much out of the information that you already have, because you have it in your heads, you have it in your medical records, or your doctors have it. And some of that information has changed over time. And so we're trying to timestamp as it's changing as well. So as we're doing this, again, Ascend is online, it's by telephone, it's by medical records, but the important thing is that you don't have to travel in terms of doing this. I'll go through the details of exactly how you do it. It is a bit of a time commitment, but we have tried to be able to make it as sort of reasonable as possible so that it didn't overwhelm people and that they could do it at their own pace. So it doesn't all have to be done at once. You can pace yourself in terms of being able to do this. Again, I'll go through the details a little bit more, but the KOALA visit, which some of you have already been a part of when we were in New York, we're continuing the KOALA protocol. So the same types of things that we're doing, we're now doing it at Boston Children's Hospital. And even though it says children's in the name of the hospital, we are not limited to children. So we see actually individuals across the lifespan, and we can accommodate individuals across the lifespan for the KOALA visit. And we are doing what I think of as, again, head to toe in terms of being very comprehensive. And I'm very happy to say we have experts here at Children's that are in some cases even deeper experts in some of the areas that we needed. And so I think we're going to be able to get even deeper assessments for KOALA. We're doing these together because they actually reinforce each other. There are some critics, not myself, but there are some critics when we go to publish this that say, well, if a parent reports something, how do you know that they're accurate? And so the reason we get medical records and we have KOALA as the in-person assessment is because that adds veracity, or that supports what you're reporting to us. And it allows other people, eventually the FDA and others, to be able to say, it squares. It looks exactly the same. So we can trust that when parents are reporting this, even at time points that are not documented in the medical record or documented during a KOALA visit, that we can trust that parents are reporting that information accurately. Within this, it's very important that as much as we can, that everyone is counted. Number one, our numbers are large, but they're not as large. I don't think we're represented in the medical literature as much as our numbers actually are. And so I think people are underestimating how big we are. And as many of you know, the number of different variants in the KIF1A gene is quite numerous, over 100. And to a certain extent, there's variability between people. And one of the things I know you ask me all the time is, what can I expect in the future? And we're developing very sophisticated computational tools. You hear about artificial intelligence and how you can use machine learning to be able to make sort of ingest large amounts of information. We're doing that with the information that we have so far so that we can answer your questions about what can we expect in the future. And that comes to bear, as we'll get to in the end, where some families are trying to make decisions about risk and benefit about certain interventions. And so we're trying to give you as much prognostic information as we can. And we may not be perfect yet, but we're doing the best we can as we do this. Okay, so next slide, I'm going to go into detail of each of the two of these. And by the way, you can participate in both of these. So if you can travel, then you can participate in KOALA, but we hope everyone is at least doing the Ascend online. And then for those people who can travel to Boston to do KOALA. Okay, next slide. All of these data, and we'll get to it again, Sarah will be talking at the end about the ASO. For individuals who eventually would get into, and I will say it's whether it's an ASO, it's whether there might be part of other treatment trials that are available in the future. And I do hope there are going to be many other treatment trials available in the future. These will provide the baseline data for comparison to be able to understand what were things like before to compare to what might be happening after. Okay, so here's the email address that's important. So if you are not already in Ascend, if you'd like to be in Ascend, if you can't remember if you're Ascend, if you think you're stuck and need to know how to go forward, all questions, one email address, one clearinghouse for everything. Dr. Rincon, who was on the screen a little bit before, she's the one who's going to be answering your emails. If you can write it in whatever language you need to get to us. I will say that, you know, if our grammar isn't perfect in your language as we write back, I hope it's close enough that you get the message, but we are trying to come to everyone where they are in terms of those languages. Okay, what are the things we're going to be getting down to? The most important is that one of the things we need to do is to see a copy of your genetic test report. So we need to make sure that you actually have KIF1A associated neurological disorder. And there's some words that get used, and I'll just explain. The words that sometimes get used are pathogenic or likely pathogenic, which is just gobbledygook for saying we think that's the answer. Sometimes there is something called a VUS, or a variant of uncertain significance. And many times what that means is the laboratory doing that test doesn't know whether or not this is the answer or not the answer. I can tell you historically, as we've done this for years, there's about 5 to 10% of people who come to us that think that KIF1A is their answer, and it's actually not their answer. And so we help them get on their path to what is their answer, but we don't want them confused, and we don't want also to have the wrong information in the natural history study for KIF1A. So we do that as the first thing to be able to come in and to make sure people are in the right club and that this is where they belong. So we do need that copy of your genetic test report. It can be in whatever language it was from, but we do need to have a copy of that. And so when you sign up, you'll sign a consent form. This is all electronic, but you'll do a consent form. And the first thing we're asking for is a copy of that genetic test report. To set expectations, as I said, this is not, let me underscore, it is not one and done, right? Your lives are not one and done, and so we're not one and done either. We need to follow you to be able to understand as things change, and we want to be absolutely poised so that if tomorrow someone comes with a new therapy, each and every one of you is ready to be able to go into that therapy if you want it. We want to have your baseline data. We want to be at that starting line. We want to be ready to go. And so this is the way that each of us can be ready to go in terms of that. And so as we're doing this, this is why we need to get updates over time, because we don't know when that might happen. And I keep hoping it's going to happen tomorrow for each and every one of you, and we want to be ready. Okay, next slide. As we do this, as I said, the steps just to lay them out for you are that the first thing, again, that email address at the bottom of this slide, I keep reminding you so that if you didn't get a chance to write it down, it's right there. That email address, right, Dr. Rincon will get back to you. Have your genetic test report, get a copy from your doctor wherever you need to dig it out underneath your bed, but get out that genetic test report. Then you're going to do the online consent. Very easy to go through that. If you can't, if that's not in your language, Dr. Rincon will go through that on the telephone with you or on the computer with you with a translator. So we're trying not to leave anyone behind in terms of this. I apologize, but we do need to do that then on the phone or online with you. So we'll have to set up a time to do that. And you might be 12 hours in a different time zone than Dr. Rincon is. So work with us in terms of being able to get you on to get this done. After you sign the consent, then we can start getting you through in terms of doing those questionnaires. Some of them, you're going to breeze right through it. Some of you have more complicated medical histories. And the more complicated medical histories, I apologize, but it'll take a little bit longer to get through with that. Okay, next slide. I wanted to show you that this is chunked up into little bits of time. And so I put the time under each one of these segments, and that each one of these, it takes a little bit of time once we get your copy of your genetic test report, because you might send it to us on a Friday. And Dr. Rincon is not working on the weekend, so it might take us a couple days to get back to you. But once we have a chance to review it as a group, then we will push you forward in terms of the consent. And then once you sign the consent, we will push you forward in terms of doing the online survey. But there is a little pause between those two steps that I said there. I'll show you in a little bit more detail, but there's a series of surveys with the online. You can stop after you finished one of those surveys. You can even stop in the middle if, something happens and you have to, get something out of the oven, but you can stop during this process as you're going forward and come back to it. And we have used these as standardized measures that things like the Federal Drug Administration or the same group in Europe, the EMA, use these as outcome measures. So we have tried to use similar instruments to what someone might want to do in a clinical trial and use it with things that we know are acceptable. Now, I will tell you there are a lot of flaws to these, but it's the best that we've got, and we are getting better. So I want to rest assure you, like, we're not stopping just with sort of things that might be mediocre. But that's why we do things like the Vineland Adaptive Behavioral Scale, and you might get annoyed by the questions. I totally agree when you get annoyed by the questions, but this is what the FDA likes doing for now. And so we're doing that to, again, be ready and at the starting line. Next slide. Let me go through with you and explain some of the things that we're starting to do to be able to get better at this. So the medical history, as I said, the medication history is important in terms of knowing what you've taken, what's worked, what hasn't worked. That's what we're getting from that to help you and help each other. The Vineland we talked about, and the seizure survey for those of you that have a history of seizure or epilepsies, very important. Within that, one of the reasons is because that may actually be an important thing to monitor in terms of whether a treatment is working. And so we want to have the most accurate information possible for that. At the end of the day, what matters is your life and overall a summation of how you're doing. And so we call that quality of life. That to me is a very important sort of composite measure about how you're doing and how any treatment might be doing. And so again, we have that as a standardized measure. Within this, we also know that there are other things like sleep that can be important, also in terms of sleep as a reflection of seizures, sleep as being able to help individuals learn. There are multiple things related to sleep, and so we have that as part of this. And then finally, we know that there are some people that are on medication for spasticity in terms of baclofen, and so we have some specific questions. I will say that as we get more sophisticated, there are things that we're looking at in terms of what I call novel or new outcome measures, things that are going to be even more granular, more detailed, but also more burdensome. And so we have not put those into this first pass because we didn't want to overwhelm people. But for some of you who are willing or interested, we'll have opportunities to go deeper in terms of these. Next slide. Within this, just to remind you, this is what we've been seeing out of the previous studies. And so any of these slides, by the way, available afterwards, so don't feel like you have to furiously write this down. But these are the frequency of things that we've been seeing for individuals who have been doing the online survey as well as the KOALA study. Within this, I think it's important that I don't feel like we know all the manifestations yet because individuals are still young and still getting older. And some of the things, although it's, I think, mostly related to the brain and to the nervous system, we do see other parts of the body affected, things that are affecting the intestine, the gut. That may be related to the way the nerves come to the gut, but we think of them as gastrointestinal issues. Obviously, vision. It's not just the optic nerve atrophy, though. We also realize that there are other issues, including cataracts, and there may be other issues. And even some, they may not, thankfully, be very frequent, but other issues that may be related to the sort of kidney and the reproductive system as well. So these are the things we're seeing. There's nothing radically different from the last time I talked to you over the summer. So I don't want you to panic. There's nothing scary that we've identified that's different or new. This is similar to what we've seen and shared before. Okay, next slide. As we're doing this, and I should just to emphasize, when we talk about medical records, one thing we've done in the past and will continue to do is to get not just reports, if you're willing to share these, but actually MRI scans themselves, as well as EEG data itself. So not just the reports, but some of the primary data, because there are some additional, more detailed comparisons and analyses that we can do that you can't just get from a report. So again, we're thinking about essentially using the power of everything that you've done before and trying to be able to get that and timestamp it over the life course, because we can't turn back the clock, but we can go back and get data from things that were done over the life course. Okay, so I'm going to switch gears now. And now this is the second part of what we're doing. This is the in-person KOALA study, the KIF-1A outcome measures, assessment, longitudinal, and endpoints. This is what you come to Boston for. And one of the things that is a little bit different from the way that we did this before is that you are going to need to get yourself to Boston. Again, correct me if I'm wrong, Angie, but right now there are not travel funds to support you coming to Boston. But if you can get yourself to Boston, then this is supported in terms of the assessments, the evaluations that we're doing. So you're not paying for this. This won't get charged to your insurance in terms of doing this. And as you'll see, this is quite a sophisticated way of being able to look head to toe in terms of the things that are going on. We intend for this to be, again, comprehensive and to be able to give us a sense of where you are now so that we know over time, are things getting better? Are they getting worse with any sort of treatment? So we want to have this information and we want to know how well we can measure it. And in some cases, I'll be honest, it may be that someone doesn't have a great day. Maybe travel was hard and they woke up on the wrong side of the bed and they don't have a great day and it's not an accurate reflection of who they are. And that's important for us to know, because if we were to do that in a clinical trial, that tells us that that particular measure might not be a good thing to test to see if a treatment is effective. So we're doing this kind of as a dry run, if you will, for what we might do if it were to come to an actual treatment study. Next slide. So as we're doing this, this is a 2-day commitment. So for most people, it takes 2 days. There might be some individuals where, for whatever reason, it might be a little bit more limited and they would only do 1 day, but in general, 2 days for this. And we intend to be able to give you back information from what we're seeing so that you see what we're seeing. Within this, there's some information that while you're there, for those of you who have been through KOALA visits, you know that you're talking to people and they're talking to you to say what they see as you're standing right there. So you're an active part of that process. In some cases, there are things that have to be scored, and so we can't do it right there on the spot, but we do a follow-up visit with you afterwards. And we will get better at being able to have those more rapid. That's something that I want to do as we're going forward. As we're doing this, there are some of these things that you do need to speak English about. Anyone with any language could participate in portions of this, but some of the psychometric testing, we have the evaluators who speak English and do this in English. Other things, it doesn't matter what language you speak, we can still do the EEG regardless of your language. So if that matters to you, I'd say there's about 75% of this that anyone in any language can do, but there are some segments of this that you need to be English speaking. And I apologize, but it's just what happens when we have evaluators who are in an English speaking country. Okay, next slide. If you've done KOALA before, we invite you to come back and do KOALA again, because we're trying to do this on an approximately 1-year basis. As we've done this, this is limited now to individuals with pathogenic or likely pathogenic variants. For the first one, we're willing to take individuals who have variants of uncertain significance, because some of those eventually with more data are defined to actually be disease associated. For KOALA, we need to limit it to people where we're certain that these are all a disease associated variant. So it's not too many people that would be left out, and we would tell you based on your genetic test report if you qualify for KOALA. Within this, the assessments are how you move your body. So that's what we call motor. Neurological is which neurologist who thinks about the nervous system in terms of evaluating things. And I'm very pleased to say we actually have some world-class experts in terms of being able to think about spasticity and neuropathy, in addition to sort of the other assessments that we've been doing. Eye conditions, we have a special a genetic ophthalmologist, and then cognitive, meaning thinking. And so those are all of the different segments that we do over 2 days, not to wear anyone out, and make sure we get a good reflection of how they're doing. Okay, next slide. Within this, just to give you some more detail in terms of what that includes, the motor assessment includes for individuals who can walk, looking at their walking and looking at it in great detail to see not just how far they walk or how fast they walk, but actually how their body moves with the walking. We also do things at looking at how they use their hands, what we call fine motor tasks. And then, as I said, because the seizures are portions of this, everyone, regardless of if they've had seizures or not, is invited to have a relatively brief electroencephalogram. And we don't have to do that like smelly glue and glue it to your head. So not like you have with a clinical EEG. We have a net and a cap that fits just right on your head and slips right off your head. So it's really quick, easy peasy lemon squeezy, and doesn't take much time in terms of getting that set up. We have psychologists doing the cognitive testing that's right sized to the person, how old they are and what their ability level is. And then, as I said, one of the things we certainly found out from the evaluations in New York are the most families, I think, underappreciated how much the eyes were involved and the different dimensions in which they were involved. And so I think this has actually been quite valuable in particular because we want to keep our kids safe. We want to make sure they can see. We want to make sure we set them up in terms of accommodations for any limitations. So I think this is an important part. As we're doing this, we are getting more granular in terms of videotaping things so that if we want to do a compare contrast, we can literally hit the rewind button. So we can be able to see from one time point to another, not just our notes, but literally play the video and compare things one versus the other. And then importantly, be able to collect blood samples and urine samples for what we call biomarkers, things that might tell us in terms of how the brain is changing over the course of the condition, and be able to measure that in blood. And that might be a good way of being able to see very quickly if a new medication is helping. The blood sample, which I'll come back to in just a second, is also important for this concept of the handles, which I'll get to for the ASO. So it also helps us with that and to be able to make cell lines if it should be necessary to make cell lines. Okay, let me go to the next slide. The blood samples. So you don't have to do the KOALA study to be able to give a blood sample. So I appreciate that for many people, especially as they're thinking about whether or not either the current or a future ASO would be helpful, they're trying to figure out whether or not they'll qualify. And we need, I'll explain why in a second, but we need a blood sample to determine that. We know it's very hard for everyone to be able to come to Boston or come to the United States to give a blood sample, so we're not going to make you do that. We do, however, need a blood sample. We can't just use a cheek swab. We can't use a saliva sample. We need to get a blood sample, and you will need to partner with a local person who can, local doctor most likely, who can get the blood drawn and then ship it to us. Again, that same email that you wrote down at the beginning, if you want to do that, Dr. Brincone and our team will help you. We will ship a collection tube and instructions out to your home. You can work with your local doctor in terms of getting those tubes filled up. It's not too much. And then we will pay for the shipping for this to come back to our laboratory and then pay for the sequence analysis that needs to be done to be able to see if you have the right handle, either for the currently available ASO or a future ASO in the future. So within this, we will, and if we, the next time we have a family meeting, we will have it set up that we will have people who can draw blood on site for that, so that if you want to get blood drawn, just like we did last summer, you can get your blood drawn in one-stop shopping, get everything done. Okay, next slide. As we do this, this is just showing you in a slightly different way, but showing you all the steps that go into this process. I hope that it's relatively easy. There's that email address again. So if you wanted to know how to sign up, there it is again. As we do this, it shouldn't take very long to get you the kit. You can do it if you're getting blood drawn for your health checkup for other reasons. You can ask them to tag along and sign this up as well. And it's nothing special that has to be done. The blood samples can be shipped at room temperature back to us. And like I said, if you're overseas, we'll include the paperwork that needs to go on with customs as well. Okay, next slide. I want to spend just 1 second because I know this is really confusing, and I know people, a lot of questions come to many of us in terms of how to understand this. So on your genetic test report for KIF1A, it will list a particular genetic variant in KIF1A that we think is disease associated. And so I've shown on this slide in green 3 different positions: P305L, R316W, and T99M. These are all 3 different mutations in the KIF1A gene that all cause CAND, and they're all slightly different, and they all have slightly different prognoses, but they are all KIF1A genes. Okay. In the ASO that was developed for the first person that got the ASO, it was for someone with the P305L mutation, but the ASO does not target P305L. So with this, it targets a different, completely benign, but genetic difference, which is next door. And that one I've called the handle. That's in blue here. Now, the handle is on the same purple strand of DNA as P305L. So when the ASO goes and grabs that handle and degrades that RNA, it degrades the RNA with the bad P305L. And it leaves that reddish squiggly there, which is the good KIF1A gene. It is specific to just one of the 2. And we need it to be specific because we need some KIF1A. Okay, so when it does it, it grabs it in a specific way, but it grabs it on that handle. I do not know who else has that handle by looking at your genetic test report, because no one thought to sequence that DNA for that handle. Who would have known? We didn't know that the ASO was going to be to that handle. The laboratories around the world, they didn't know we were going to do this. So they didn't sequence it. They didn't report it. It's not in your genetic test report. It's not in your medical records because it is buried in a place that they did not sequence. So we have to proactively go out and sequence afresh to find out who has that handle A. And not only who has that handle A, but who has it in a configuration that we call cis on the same copy of the DNA squiggle as your particular disease-associated mutation. So that when we grab the handle, we don't degrade the wrong one. It would be the worst thing in the world to degrade the good KIF1A and leave the bad KIF1A, right? We don't want to do that. So we have to be very careful that we know how this is configured as we're doing this. In doing this and looking around for round numbers, I would guess it's going to be about 10% of the community where this ASO is going to work. So it's not going to be 90%. It's not even going to be 50%. At the end of the day, if we're going to use an ASO strategy, we're going to need more than one ASO for many different handles to do this. But the good news is I don't think we're going to need 100 different handles for 100 different mutations, right? So the amount of work that we're going to need to do is going to be much, much less if we can grab a few handles and use those across the community. And we could talk more about this, but that's the basic concept is you don't know your handles. I don't know your handles unless we did a specific experiment to now understand your handle and your configuration. Okay, I hope that makes sense. Next slide. And that might be the end of mine. Yep, I think that's the end of mine. And maybe Sarah, we're going to do the handoff here.
Yeah, thank you, Dr. Chung. That was great. And I'd like to introduce Dr. Sarah Glass from n-Lorem now. She is so gracious to join us today as well and present information that we've all been following along with. And we'll take it over from here if Sarah's ready.
Yeah, great. Thank you, Angie. I also have my colleague Lori Mignon on the phone here. She leads clinical development, so she spent a lot of time with Wendy and Jen Bain and others, and of course Luke and Susanna. So, so just wanted to share, Wendy, thank you for that really robust and very specific description of the ASO. It sounds very complicated, but ultimately, Wendy's taking full care of genotyping, identifying whether you carry that particular SNP, and then being able to evaluate in the future what others might be able to, we can potentially target what other SNPs that we might be able to target with different ASOs. So ultimately, the current trial is with a single patient. And so that's with Susanna Rosen. The key here is that there's a lot of data. And so one of the critical pieces in an individualized treatment is that that individual is their own comparator. So collecting the data that Wendy just described, the natural history, enables us to be able to know all of that natural history about yourself or your loved one that is afflicted with this disorder. And once we start treatment, to then be able to continue collecting the data and to see the effect of that actual treatment. So here, there's obviously a lot of parent-reported data. This is on the seizures, very specific and detailed information on seizures, how many, the different length, falls, and quality of life. And ultimately, what we are seeing in this trial and with Susanna is a lot of positive results. So, decreases in the seizures and the length of time of the seizures, a decrease in the number of falls, an increase in quality of life. Wendy and her team have drafted a manuscript and should be coming out as well. And, like was just mentioned, is that we do believe there are going to be a number of other patients, and we've already identified one patient who also carries this particular handle that is on the proper allele that is with respect to the patient's mutation that has been accepted into the n-Lorem program, that we have already moved forward on that regulatory path to be able to treat that patient. And so again, this is using the same ASO. We know that not every patient will be able to be helped with this ASO. And we also know, I think it's important to highlight, and we say this at n-Lorem all the time, is ASOs are one of many really new innovative therapies. And so, really being able to target these mutations and think on the genetically focused therapies, ASOs are one, and unfortunately won't be able to help everyone. So n-Lorem on the next slide is a nonprofit organization. We were founded in January of 2020 with really the goal of saying, individuals who carry these very, very rare mutations don't typically have an opportunity to have drugs discovered and developed for them from a commercial perspective. And so what we do is that we create ASOs. So in the case of Susanna, it's ultimately evaluating her specific genetic profile to say, here's what the mutation is. How can we actually understand this sequence in a very detailed way to be able to discover and develop a treatment that can potentially help and have some positive clinical outcomes? So I think importantly, this is an investigational treatment. These are under a very specific regulatory path as defined by the FDA. These are individualized research INDs, individualized ASOs. And so ultimately, what that's going to enable for individuals who carry that handle is that this will be a more efficient path to a treatment because the drug itself is in hand. The key piece here is that we still, after identifying each person who carries that particular handle, there are a couple of other steps that we'll talk about in the next couple of slides, just to highlight that before we can actually get to the position of being able to leverage that treatment at this point in time. So I think it's important, really, one thing we're trying to do, and especially to support the patients and the communities, to continue to acknowledge and applaud and commend our physicians and our institutions for all of the work and the support that they do here. So the key challenge is that for many of these physicians, this is kind of something that they've been doing more just out of passion for wanting to help you and I and us. This is not something that the institutions often can say, okay, we're going to give you every Wednesday half day to focus on this. No, this is something that they do literally nights and weekends and really doing everything they can to help us. And so it's really important upfront that we are in contact with your physician. And if it's Dr. Chung, obviously we've worked with her a lot, to make sure that we're able to ensure that you can actually get the treatment. Once we do all of this lead time, all of this work in advance, to then get to the place where an institution says, sorry, we're not able to help. We don't want to get there. So that's why we're stressing at this point in time to say, you coming to n-Lorem in partnership with a physician, Dr. Chung or Dr. Bain or other neurologists, it's a really critical piece because we have to work with that physician to be able to get this treatment forward. So I think there's a couple other things on the next slide that we wanted to really highlight about the process. So in addition to this initial, genome sequencing that is going to really highlight this variant, this handle, as Wendy is calling it, We'll also need this iPSC, a particular cell line that's going to be required for each patient. And n-Lorem can work with the physician when that doesn't exist to really try to acquire those cell lines. And then ultimately, in order to evaluate, what we'll do is to evaluate having that handle, looking at the cells, testing the ASO in the patient cells to ensure that we see the effect we're expecting. And if that is the case, then we will go through that regulatory submission. So like I said, you can see that first sub-bullet. That's all on the clinical site startup as well. And the treatment's not guaranteed, but we are, fully vetted in this process. We've submitted 11 INDs across different mutation-specific disorders. We know very well how to navigate this system, and we're going to do everything we can to work with each of you to make sure we can do that. On the other hand, for other individuals who still carry these mutations that are in this nano-rare space that don't really typically have the opportunity for commercial development, we still are very much open to and interested in trying to evaluate whether we could understand if we can help. And so that takes an application, again, with a physician, to be able to then enable us to look at that sequencing, understand if we can identify a region that we can bind an ASO to be able to have that optimal effect of an ASO to knock down the KIP1A. And so, at the end of all of this, if we're successful, it's n-Lorem's commitment. Each drug program costs us about $1.5 million to US dollars to go from patient mutation to drug in hand for the physician. And it's our commitment to ensure that the physician has enough drug to treat each patient for free for life, as long as the physician sees benefit. So I think a couple other specific things we wanted to just touch on, and Lori, feel free to chime in here too, but just again, so that there's, full awareness, honestly, of what to expect, because I think in the last 4 years that we've been in existence, we definitely acknowledge that sometimes people will start to even have conversations with n-Lorem and say, okay, well, where's my, pill I'm going to take, or where's my first dose, or some of these questions. And that's on us for not doing enough to ensure that we give the information at early enough time. So really, this is it's an intrathecal injection. So this is a spinal injection. This requires an overnight stay at the hospital for every dose escalation, and a lot of monitoring as well. And that's really important because, the safety of each individual is of the #1, priority for us as well. And the cost, like I mentioned before, on the entire drug, that's all incurred by us. So nobody will ever see any cost associated with that. And the cost of the treatment, delivery, and evaluation are variable, and those are highly dependent upon the institution. Let's see, I think we had one more slide. So when we're, I think in addition to the genetics, on the front end is really trying to think about how can we, in general, n-Lorem, we have a bias to treat. We're trying to help as many as we possibly can using this nonprofit model. We're really and truly trying to focus on the risk benefit. And this is really critical for your physician to have a very solid understanding of sort of the clinical profile at the time of application. It's also around thinking about the physician themselves. So really thinking about, is the institution able and willing to be able to support this? And Dr. Chung and Dr. Bain both listed here as key leaders and wonderful supporters that we're already working with, and we know we have a path forward with them. So I think the last piece here we'll just mention, we have a pre-submission process. So we do have a number of individuals who have a mutation. The question is, do we think and this is not a process that's going to go outside of actually doing the sequencing for the handle that you have to do through Wendy. But this is if there are mutations, there are questions on whether you think your mutation may be amenable at all to ASOs. So there are some key categories of mutations that are not amenable to ASOs, like null mutations, for example. But there are many that are more common that we think if we say through this pre-application process, we can at least try to help rule out any that we don't think we can help to be able to save time. And that will really enable you to say, okay, ASO is not for us. Let's work with our physician and others to try to go down those other paths as well. So really trying to think in a way that will help you get to the best therapy for yourself and your loved ones as efficiently as possible. I think that was it, right, Angie? If, we're happy to
Yeah.
answer any questions. I know there were a lot in the chat, so I don't know if you want to pull some of those up, but,
Yeah, thank you so much, both of you, for all this wonderful information. I think there was one more slide before we go to Q&A, and everybody should laugh because this is our flow chart. Dylan and I were talking about, well, how simple it all is, right? Anyways, you don't need to panic and try to figure out where you're at on this flow chart right now. We will share this with everybody and get it out there, but you're basically entering in 3 different ways. If you want to know about the handle, you know your result, it's either yes or no, or you don't know. So we can talk through this more, but n-Lorem, Chung Lab, Boston Children's is all in here, and everybody can find themselves someplace in this crazy diagram. So I think I'm going to stop sharing with that, and Dylan is going to help us facilitate the Q&A.
Yeah, so I wanted to start with discussing some of the factors for koala participation. In particular, we have families wondering if there are any steps that they need to take if they have participated in KOALA at Columbia now that you've transitioned to Boston, whether there are any other I's they need to dot or T's they need to cross.
Yep. So all of the data has come over from Columbia to Boston, so we know who you are, and will continue seamlessly in terms of doing that. The measures that are comparable, we're doing exactly the same measures, so the data will be comparable. Mostly what we're doing is going deeper in ways that I think are going to be more meaningful, as we've learned from the first phase of this. If you're interested in coming back, then again, send an email to that Ascend email address. We're not assuming that you want to come back, but we're hoping you are. I'll also say, we had the family meeting, and I hope we continue to do this, where we can be families can get kind of a 2-for-1 essentially by coming to a family meeting and doing assessments in person there, and hopefully minimizing the burden. And I hope, my own personal hope is that we can actually travel to multiple places to make this more accessible to more people as we do it. I don't know exactly in terms of commitment where the family groups are going to go, but we will try and meet you and go where you are. I can't hear you, Dylan.
You're on mute, Dylan. I have a question. If you're having audio problems, I can jump in with it. I see a lot of in the chats about and just in talking to a lot of families about the issue of international application. And I think that it's important for people to understand, and maybe Wendy and Sarah could comment on this, that it's not a choice made by the institution or n-Lorem. It has a lot to do with the regulatory pathway, and if that's true, and how our international families should
I'll let
proceed.
Sarah talk about this, because this is on their application.
yeah, so we're, right now the regulatory path exists. It's a very specific, individualized ASO guidance documents in the US. Those exist. They do not exist elsewhere in the world yet. That we have been in conversations with many regulatory agencies in a number of different countries, and there is absolutely a willingness and an interest in making this happen. A lot of times, many countries will look to the FDA to see what the guidance documents are there. And so I think it does look like there will be progress as far as what those look like. From the n-Lorem perspective, is that if we feel, and if, based on this genotyping that Wendy does, if there is a potential to treat someone in a different country that with the ASO that we already have an approved research IND for, that is a path we are, at this point in time, potentially able to explore and pursue. And so that's what we, I think we've talked about. We're not able at this point in time, if someone does not have that handle and cannot be treated with that ASO, to say to someone in a different country, okay, we're going to start from scratch, because we don't actually know what the path is that they would require technically. But what we can do is to go to a different country, potentially, if that individual has a physician supporting them, to say, here's an approved FDA research IND, and here's the treatment. and of course, see if we can navigate a path. And I think the most important thing here is we haven't done this yet. So we'd be very much working with each of you to see what we could do. Adrian, I
Thanks.
don't know, it looked like you had your hand up there.
My niece, who's the person who has this, is an American citizen but resident in Canada. She's got 3 nationalities, Canadian, British as well. So would she be able to access this and go to Boston and do the tests, or how would that work?
Yeah, I mean, it's for us, you know, honestly, wherever one is a citizen, it's really focused on the fact that we can get a drug through with a US regulatory path. And so if one, you know, has the ability and has a physician support to be in the US, absolutely, we can work with that patient and their family in the US. I think, again, if this is with the ASO that exists now, potentially, you know, Canada and UK could be an option as well that we could.
Yeah. I mean, I'm just, you know, curious as to, because she is an American citizen.
Mhm.
And
yeah, so really it's all about the physician here. So I think that's what, you know, I would just, if they have, you know, if she has a physician that could come to us and we could have a conversation and you know, Lori chat with that physician.
Okay. And the other thing, ASO, what does ASO actually mean, this acronym?
Sorry, antisense oligonucleotide.
Antisense.
Lori, do you want to talk a little bit about antisense and a little history?
Yeah, they're called antisense oligonucleotides. And so they're little pieces of DNA that we put together that can bind to the RNA. So it's little pieces of nucleotides. So using the same, you know, 4 letters that we have in our DNA, A, T, C, G, and we just figure out the right sequence to bind to that correct spot on the mRNA. And so, yeah, we call them antisense oligonucleotides, ASO for short, because it's a mouthful, but that's basically what it is, nucleotides.
So what you're trying to do here is find things that will, you know, lock on to whatever ASO the patient has.
So the patient just has the genetic code, and they have a mutation or a SNP or handle in that code that we can then develop our ASO against because we know the sequence. Yeah.
So it will lock onto that individual sequence that they have.
Yes.
So it will work specifically for them, wouldn't work for somebody with a different sequence.
Exactly. Exactly. And what it will do is it will bind specifically to that sequence and that hybrid, because the cell doesn't like to see those double-stranded pieces there, will actually cause the degradation of it. And it will cause the degradation of it specifically of that just one side, not the other one, so that you still get wild type protein being made.
I'll back up to a little bit more about enrollment and just some practical questions that people had. If somebody is unsure of where they are in terms of completing Ascend or KOALA information, are there any touch points that they can use to assess how far along they are in all these processes?
One email address. Just send an email to that Ascend. We'll let you know where you are, what you need to do next.
Great. And then do you have to complete KOALA to be eligible for clinical trials?
From my point of view, someone needs to have a baseline to know for comparison. You technically don't have to do KOALA to do that. If you were doing it with us in Boston, we're going to have you do basically KOALA. So again, my thinking is you might as well do that now to be able to make sure you're ready. And I would say that's whether it's for the ASO or for other things. But no, if you're going to do this through someone else and they don't need to have KOALA data, then you don't have to, but I think it would help.
And do KOALA participants receive any results from their assessments, things that they can look at with regards to their symptomology?
Yeah, I tried to say this during the talk that I gave, but there's literally real time where we're talking to you during an assessment, and then there's some that require scoring that we do after the study visit is over. But yes, and people have gotten pictures in terms of their eyes to take back to the ophthalmologist. We're trying to make this as useful to you as possible.
moving on to some of the ASO questions. The first that came up was now that many blood samples have been assessed, do we have a better idea of how many handle-based treatments would actually cover our patient population, or how much this would need to get scaled?
So I would say that to get the vast majority of people, we're going to need at least 10 different ones. The question is going to be, which I don't know, is can all 10 of these handles ideally actually be targeted? It's not clear that they're all actually going to be able to have an ASO developed for them. So it's a matter of experimental data to say whether or not that's the case and how to go forward. But it's not just going to be 1 or 2 or 3.
And then with regards to actually administering the ASO treatment, if somebody already has an existing intrathecal pump, is that something that could be used as an administration method, or do these need to be 2 separate protocols?
I can take that question if you want, Wendy.
Go ahead.
Yeah, so normally, I mean, the injections with the ASOs have to be intrathecal bolus injections normally. So there are limitations if somebody, let's say, has a shunt already placed, then we would have to talk to the physician to see if we could still do an intrathecal injection and if the shunt can be turned off for a certain amount of time, because it's really, and the reason why we do intrathecal bolus injection, it's because the bolus push actually pushes the drug and allows for diffusion and, of the ASO all up the spine and into the brain tissue. And so if a patient has a shunt or has anything else, we just need to make sure that we discuss this with, the right physicians to see if ASO treatment or intrathecal injection can be done as well.
Thank you, Laurie.
Mm-hmm.
I saw somebody asked how often are the injections, and I think that's still kind of being determined in Suzanne's case, right?
It is, but in general, for most of our intrathecal programs, I can say that, you try to increase the drug level in the brain over the first 3 months. So you inject on the first, 1 day, and then 1 month later, and then 2 months later. And then after that, it should be every 3 months. That's about the frequency. But it is true that it will also depend based on the response of the patient, the clinical response, based on safety. So obviously safety is always the most important thing. So, we'll be conservative at first to make sure that that we're on the safe side.
And I think that's a great segue into our final point. I want to be respectful of everybody's time, but somebody asked just more broadly, how should they be thinking about risk and reward with regards to their family member and the, I guess, commitment that an ASO trial entails? That's a philosophical one.
I would say it's a very individualized decision, and you have to think for yourself in terms of what you feel that the burden is in your family and for the individual in your family. Number one, this is not a one and done treatment. I think that's good and bad. I think the good is that it's not a forever thing. And if there's a problem and a complication, it washes out of the system. The bad is that it requires repeated administration. So this is not done. So that's what we have right now. I do hope and I do fully believe there are going to be other things beyond the ASO, but they're not here today. And so it's going to take some time to evolve. And there are a lot of people working very hard in some of the solutions across neurological conditions, not just for KIF1A, I think are going to be very helpful to this community. And so we're going to ride the wave of many other rare diseases as well, and rare neurological conditions. And I am just knowing some of the things that are coming down the road, I am hopeful, but it's not today. And so people just going to have to make their own decisions, and it's a hard decision.
And actually one last question, because it's just very straightforward. If my child had SDR, can they get involved? Is a spinal dorsal rhizotomy a consideration for people wanting to get ASOs?
I'll say, if the question is geared to me, that we have actually at Children's looked at some complicated cases, and I think we've got more options in terms of delivery, but they're each individually reviewed, and we have to have images, we have to know medical history to know from a technical point of view what's possible.
Well, yeah, thank you, Dr. Chung and Dr. Glass, for sharing so much information about this. I know that we have many questions, probably additional questions that we still need to get to, and we've recorded that in the chat to share with you. We'll also be sharing this presentation and slides for everybody so that everybody is on the same page with regards to these updates.
Thank you, everybody, for attending. We'll, like Dylan said, we will get all this out to the community to review the slides, the questions, and maybe we can engage again a little bit more with Dr. Chung and Dr. Glass, Dr. Minion on any questions that we didn't get addressed. But with that, I think we will close out our meeting and appreciate all of you for attending, and especially our presenters. Thank you guys so much. We couldn't do any of this without you.
Angie, can I just say one thing is that you know, Sally and I are going through it and are here for anybody to call or just send us an email, or you all have our phone numbers, and if you don't, you have our emails, but we're here to help and answer questions, and just we're so thankful for n-Lorem and Wendy and everybody and all of you, so thanks.
Thanks, Luke. Thank you, everybody. Have a good rest of your day.